Yeah, okay, at your convenience.
All right.
Gentlemen.
Well, most people in here don't, but so I'll introduce you. So this is a final grand rounds for 2013, and our speaker today is Yiqiang Zhang from Health Science Center. He's in the Physiology Department and Barshop Institute. I've known Yiqiang for a while now. We both were doing postdoc with Arlen Richardson over the last 5 or 6 years now. So he's now got his own set of research, focusing mostly on tissue and cell modification, primarily through oxidative stress, but things like cell senescence and how they affect both aging and age-related diseases like sarcopenia. And so I think I'm gathering from the title of your talk, you're going to talk to us about SOD1 knockout mice, how oxidative stress can drive sarcopenia, which is going to tell us why that's important. So thanks, Yiqiang.
Okay. Thank you for the invitation. Nice introduction. Our group in the last several years are concentrating on the oxidative stress affecting aging phenotype like sarcopenia, which is has been shown to be very important for the healthy aging in the aged population. So while we are studying sarcopenia and so first, we introduce the concept of sarcopenia and some of the what we know about the sarcopenia.
Is that the one, or this is the one?
This,
Which one is it?
It's working.
This one, okay.
so sarcopenia is actually the Greek means called poverty of flesh. Sarc means the meat, penia means less, or something like that. So the definition is like this, as the in the Gary and Simmons, both they define the sarcopenia as age-related loss of muscle mass and the strength and function, which the loss of muscle mass we commonly call it muscle atrophy. And, so here is the picture shows the age associated decline of the muscle in a person. This is at age 25 and compared to age 63. And this dark area is the muscle. The surrounding area is the fat tissue, and in the center that's the bone. So after approximately 40 years in the mid-60s, you'll see there's a dramatic decrease of the muscle mass, and there's a dramatic increase of the adipose tissue in the muscle, and also the structure of the muscle change compared to the younger person, and also there's the infiltration of adipose tissue into the muscle tissue.
Can I ask a question? Is this the same patient or different patient?
This is-
Show it.
This is different.
Okay.
different.
Because I'm a little bit scared. I'm in the age.
Actually, the aged one, your picture very much looks like what the DVT mice have.
Diabetic
Yeah.
mice. Yeah, the diabetic mice has also the disease associated muscle atrophy. Yeah, in diabetic patients is also common. Yeah. Muscle loss and decrease of fat. That's more probably related to the inflammation and other factors. And the onset of the sarcopenia, this is purely only the age-related muscle loss. I'm not talking about some of the disease associated, like cancer associated muscle loss. It's called like Kakashi. This is the purely age-associated muscle loss. The onset begins at around 40 years old, and with get to the late advanced age around 75 years old, accelerated. And the most affected muscles are the first twitch fibers. As most of you probably already know, the muscle composed of two types of fibers, the fast twitch and the slow twitch fibers. And the fast twitch fiber mainly use the glycosylation, provide energy, and react very fast for the sprinters. The sprinters have a large portion of the fast twitch. For slow twitch fibers, mainly use the mitochondria oxidation provide the ATP, the energy, which have a slow response, so that's called slow twitch fiber. So the sarcopenia affect the fast twitch fiber much more than the slow twitch fiber. And there are multiple causes of the development of the sarcopenia, includes physical activity. So if the older people are bedding inactive, after a while you find there will be a dramatic decrease of the muscle mass. And also the motor unit degeneration, that's common called degeneration. And also there's a decreased hormone levels like the IGF and other sex hormones. And also decreased protein synthesis in the old people, related to the muscle atrophy. And also, luckily, the sarcopenia can be partly preventable. And one of the preventable interventions is the exercise. So you keep exercise, you will at least delay the progression of the sarcopenia, the muscle loss. So why we talk about the sarcopenia? Why we study sarcopenia? What's the consequences? So there are multiple consequences along the development of the sarcopenia, the muscle loss. So the first, you have the loss of the muscle mass, as shown earlier in the picture. Then you lost the muscle strength, so the muscle fiber decrease. So includes the number of the muscle fiber decrease, also the mass of the individual muscle fiber decrease, and that will lead to the loss of the muscle strength. And further on, will decrease physical activity. You need some kind of strength and support. And later on, you increase the risk of fall. So there are old people, you have more muscle loss. And furthermore, you lost autonomy. You need some kind of assistance to stand, to walk. And this, clinically, commonly, you can lead to some kind of called a phenomena, or this, I don't know, it's kind of called disease or something, it's frailty. So, and this leads to the dependency of the old people, not necessarily patient. So, all of these contribute to decline of the health aging. So that's the overall consequence of this sarcopenia. So as I mentioned earlier, the sarcopenia is multifactorial. Here is the most of the known factors that contribute to the onset and development of the sarcopenia, and includes the hormone, decrease of the hormone, the growth hormones, and increase of this hormone, which leads to the muscle atrophy, contribute. And the lifestyle changes, the activity things, and leads to fiber size decrease. And also the immunological factors changes, mostly the inflammatory factors increase, includes the IL-6, IL-1β, TNF-alpha, all those kind of inflammation leads to the muscle death, fiber death. And there is also the most important thing is the motor neuron death, leads to the denervation of the muscle, so leads to the muscle, the disconnection between the motor neuron and the muscle leads to the muscle won't receive any signal from the neuron, so they stop functional problem. And some of the muscles will gradually die because of the lack of the motor neuron connection. And I will focus on in the talk about this part of the factors that lead to muscle atrophy. That's the focus of our group in the past several years, which we try to understand why the motor neuron died during aging in the advanced age, which leads to the muscle atrophy and the sarcopenia. So in the process, we found that oxidative stress may play an important role leading to the dysfunction of the motor neuron and the destruction of the neuromuscular junction, which leads to the muscle fiber decrease, number decrease, and mass decrease, and leads to muscle atrophy. And so talking about the motor neuron, so the loss of the motor neuron is called denervation. So during the aging, there is a gradual death of the motor neurons, leads to the denervation. What is denervation? So it's age-related change of the neuromuscular system, may play a important role in the onset of sarcopenia. And here shows the cartoon shows the neuromuscular junction where the motor neuron connect. This is the spinal cord with the neurons, and this is the motor neuron connect to each individual muscle fiber. Each fiber have one of the motor neuron connected. There is this part called the neuromuscular junction, where the motor neuron connect to the muscle fiber. This also later on we'll talk about the end plate. This is also called the end plate, which the neural cell from the synapses connect to the muscle. The muscle part is called the postsynaptic. This is the compartment. The neuron will release the mainly the acetylcholine, and the muscle will have the acetylcholine receptor. And once the neuron releases acetylcholine, the receptor binds the acetylcholine in the muscle, will stimulate the muscle to move, contract, as the force produced.
So only one neuron connected with one muscle fiber, or there are more?
Yes, for each muscle fiber, individual muscle fiber, only one motor neuron connected.
Can the structure easily be seen under the microscope? Or?
You use a special staining. Later on, I will show you the picture. Use double, triple staining. You stain the neuron part and stain the muscle part. You will see some of the picture looks beautiful. And you need only one motor neuron to one fiber. Otherwise, you will have trouble. If you have two neurons, they are not in synchronous, you have trouble to move. So the denervation is here's the definition of the denervation. It's the number of the spinal cord motor neurons and functional motor units decline with age. So resulting from the denervation of the muscle fibers within the motor unit, which causes the muscle fibers to atrophy and may eventually die. So here's the denervation shows in the aging, and that's the when it's healthy in the young healthy adults. So each individual connects to their muscle fiber. Here's the structure you will see is very intact and looks good. During aging, you will see cover here. You will see some of the motor neurons already gone. Some of them are gone like this. Some of the and the motor neural structure start disrupted. This you see fragmented, covered here. This integrate. And during this process, there is also like muscle fiber, like remodeling, motor neural remodeling. The neuromuscular junction start from the because the first twitch fiber is easy to die first, atrophy first. So there is a model. Once those fibers gone, they start to get to the slow twitch fiber, and the slow twitch fiber are much the renewed, the redifferentiated fiber is much smaller, and the reconnection of the motor neuron is less integrated compared to the young adults, and those fiber motoneuron, reconnecting neural fibers have less force and function much weaker than the younger adults. Even you will see in the aging, you still have some those survived re-innervated new motoneurons. Those fibers not function as good as the young adults. So that's why the atrophied muscle get much weaker. So we are interested, our group is interested in how oxidative stress affect the motor neuron and the muscle. And in the to further explain that, I need to introduce a little bit about the oxidative stress. Here's the diagram shows what is oxidative stress and how oxidative stress occurs and leads to the damage to the cell. So oxidative stress is a state that's due to the increase of the production of reactive oxygen species. So we are all aerobic organisms, and the cells use the oxygen as a fuel to produce ATP. During that process, you will have byproducts of reactive oxygen species, such as superoxide. So that's most of the time, this is the first step. You generate the upstream step, you generate superoxide. Superoxide is the exit time is very short. They are converted to the hydrogen peroxide, usually by the superoxide dismutase. And they can also detoxify by the quinone system, and there is a non-enzymatic system through the P450 and the quinone system. And the hydrogen peroxide can further detoxify to water through the GPX, GSH peroxidase, GPX enzymes, and other kind of small molecules like the GSH glutathione. And also another important enzyme which located in the peroxisome is the catalase, which also reduce the hydrogen peroxide into water. However, during the superoxide and the hydrogen peroxide can go through the Fenton reaction through the ions like iron. They can produce a highly toxic oxygen hydrogen free radical. This can damage, lead to macromolecules in the cell like DNA, proteins, and lipids. Once the macromolecules damage in the cell, they lead to malfunction of the cell, can lead to cell death, like apoptosis or necrosis. And that's the general mechanism of how oxidative stress leads to oxidative damage and cell death. So the evidence that support our hypothesis that oxidative stress during aging leads to sarcopenia is this. The first is oxidative stress and damage increases with age. It has been shown in multiple tissues, including muscle and neuron. Second, oxidative stress leads to inflammation. That has been shown in multiple studies. And also, oxidative stress induces apoptosis of myocytes. And finally, because oxidative stress, mitochondria is a major source of the oxidative stress, and there's also a major source of the generation of the superoxide through the oxidative phosphorylation. So to understand the oxidative stress during aging and its relationship with the sarcopenia, luckily we have a wonderful mouse model. So it's hard to do this in human beings. So it does need a big proof from the RRBs. But luckily we have a very good mouse model, which is called superoxide dismutase 1 knockout model. What is superoxide dismutase has three isoforms in the humans. There's superoxide oxidase 1, which is cytosol localized, also partly localized in the intermembrane space of the mitochondria. And the superoxide dismutase 2, which is solely localized in the mitochondria. And the superoxide dismutase 3, which is extracellular in the fluid outside the cell. And superoxide dismutase 3 knockout mouse can survive, no problem. Superoxide dismutase 2 knockout cannot survive to die embryonic lethal. And the superoxide distributed 1 knockout can survive, and that's good for our study. And before we go to the did, we'll show what the superoxide distributed is. I already talked about this converse superoxide hydrogen peroxide. It's a major antioxidant enzyme in the cytosol, and I talked about the localization. And importantly, SOD1 mutation leads to the ALS, the clinical syndrome of the ALS, familial ALS, and which is the symptom is the neuromuscular loss, dysfunction, and loss of muscle. And in these patients, there's elevated oxidative stress and damage, indicate the SOD1 may play an important role in the neuromuscular function. So the SOD1 knockout mouse has been generated in California by the Epstein's lab group, and it's been shown that these mice, once you have these mice deleted, we've deleted the SOD1, you have increased oxidative stress and oxidative damage. These 3 parts shows the oxidation of the protein, lipid, and the DNA. And this is only shows the knockout, SOD1 knockout mouse with age for the protein oxidation compared to the ratio to the age matched wild type mouse. So in the young, there's like 100%. There's no increase or dramatic increase of the oxidative damage compared to the wild type. In the 18 months, you have a dramatic increase over like a 200% increase. And in the end stage, further increase. That's protein oxidation increase. Also, lipid oxidation increase in the SOD1 knockout mouse compared to the wild type. In each age, there is an increase from 20% from like, this is the, this is called the proximity. This is the absolute value. This is the nanogram, the oxidized lipid per gram of tissue. Also, there is a dramatic increase of the DNA oxidation. It's the OxiDG, only one of the nucleotide oxidized measure. Usually HPLC. You see this is the SOD1 knockout, significant increase come to wild type. And if you only delete 1 copy of the SOD1, there is no significant increase of the oxidative stress. You need to knock out both copies of the SOD1. And interestingly, these SOD1 mouse live a little bit much shorter than the wild type mouse. This is the SOD1 knockout mouse. They live only up to 25 months of age, the maximum lifespan, compared to the wild type and the heterozygous knockout SOD1 mouse. So our group in the like 5-6 years to further investigate this mouse model and showed that there is a dramatic decrease of the skeletal muscle mass in the SOD1 mouse. The yellow indicates SOD1 knockout mouse. The blue indicates wild type. You see, start around 400 days, the skeletal muscle mass starts in decrease compared to the wild type mouse. That indicates there is a potentially accelerated muscle loss in SOD1 mouse. So use this mouse, we can do experiment much quicker rather than use wild mouse. Wild mouse has muscle atrophy very late in the stage, normally after 30 months of age. That takes a long time. And for this mouse, we can only like after a year, around a year, the stage dramatic decrease the muscle mass. So this provides good model for short time, so we can do the experiment in relatively short time.
What did that explain that after a year there is a dramatic decline? What do you think explains that after 400 days there is a faster decline than from the initial year?
Because of the knockout of the SOD1, the oxidative stress. I will show you later on the data to support why these mice has accelerated aging. phenotype, the muscle loss compared to wild.
But you didn't change any other variables, it's just the SOD1.
Yeah, just change one gene.
I see.
Just change one gene is because it's dramatic phenotype
Okay.
in the muscle. And he shows the real tissue of the muscle, wild type, and the SOD1 knockout. In each of the gastroc, TA, or the EDL, or soleus, or percept, each of them are smaller compared to the wild type.
Yiqiang, what is the fast twist muscle in one of the?
Yeah, there is each of those different tissue fiber has a mixture of fast twitch and slow twitch. Yeah.
Okay.
The slow twitch mostly are much darker red.
Red,
The fast twitch a little bit whiter, but the fast twitch has two types: type 2A and type 2. Type 2A and type 2B. Type 2B a little bit closer color-wise to the slow twitch, a little bit red. The type 2A fast twitch fiber is a little bit white. It's like in the muscle fiber, if you dissect the left, you will see some white, very white one. Those called fast twitch fibers. But this all mixed is a mix. Here indicates the whole mouth, the hind leg is white. This is the it's hard to see. There's you can see some decrease of the muscle compared to the wild type. And if it shows clearly, and there is a little bit darker, redder compared to wild type, that means there is more loss of the fast twitch fiber in the SOD1 mouse compared to the wild type. That's very consistent with what we observe in human and other models for the sarcopenia, which the fast twitch fiber goes away faster during sarcopenia. And first we try to see how to prevent the accelerated loss of muscle fiber in SOD1 mouse. We use this non-genetic approach called dietary restriction, and which has been shown to extend the mouse lifespan and also other extend lifespan in other species. So first we show that the SOD1 mice on dietary restriction has a slower decrease of the muscle GT, that's the gastric image. Compare this wild type knockout is knockout on dietary restriction. So for we look at the younger, middle, and old ages, in every age we see a increase of the muscle fiber after the dietary restriction. That indicate that dietary restriction is very beneficial for the muscle loss. So here shows the dietary restriction also dramatically improve the structure of the muscle fiber with the Z line, something like that. This is the fiber. You see that the mouse has disrupted Z lines and very compared to this, there's so many different changes. And once this mice on dietary restriction, we start close to the wild type. That's dramatic.
So what are you restricting?
This is like 40% restrict on the dietary restriction.
In terms of what? Calories, protein?
No, we don't consider calories. We call it dietary restriction instead of calorie restriction. It's just a diet. We put the mouse on a certain period on normal ad libitum to find out how much they consume. Then you reduce those to 60%.
So it's smaller portions.
Yeah, just get rid of 40% of the food. Yeah, just a whole food. So we don't know how much really they take the calories. So we don't call it calorie restriction. There are probably multiple factors, includes color restriction, protein, some of the amino acids of those types. So besides restore the muscle mass and improve the muscle function, it also reduce the oxidative stress in the muscle, liver, and brain, the SOD mice after the diet restriction. So this is only the lipid oxidation matter in these mice. So in our tissue, after diet restriction, they all reduce. Oxidative stress all reduced. So that's one hypothesis that we propose that diet nutrition improve the muscle function and mass in SOD mice, probably through the reduce of the oxidative damage. And also diet restriction increase the lifespan of the knockout mice. This is the knockout mice. This is the wild type mice. This is the SOD knockout mice on diet restriction. See, their lifespan improved dramatically. And this is the wild type mice on diet restriction. That's a 40% increase in lifespan. However, the diet restriction on the SOD1 mice did not further increase the lifespan beyond the wild type mice. Why? We don't know that. We'll still investigate what's the reason behind this. So we ask why the deletion of SOD1 leads to muscle atrophy. So the hypothesis is that SOD1 loss leads to the increase of the reactive oxygen species, and this includes leads to the oxidative damage, and which further leads to the degeneration because of the death of the motor neurons, and leads to the muscle loss, the death of the muscle fibers, and also leads to the function deficit. So one of the things coming to our mind is because the neuromuscular junction composed of two compartments, one from the motor neuron, the other from the muscle. We want to know the SOD1 loss in which department contributes more to the muscle loss. Is the neuron or the muscle fiber, or they need both? So to answer this question, we need to use a genetic strategy. That strategy is to knock out SOD1 specifically in neuron or specifically in muscle. Another thing is to overexpress, to restore the SOD1 expression either in neuron or muscle of SOD1 mice. So two strategies. One is in wild-type mice, you knock out either in neuron or muscle. The other strategy is restore the SOD1 in SOD1 knockout mice, either in muscle or neuron, to see what happens to the muscle function and muscle mass. So this is the strategy one. We use the tissue-specific knockout mouse strategy. So we constructed a flux mice, use this construct, which has a pre-log system inserted into the SOD1 gene locus in the wild-type mice. So these mice behave and the expression of the SOD1 same as the wild-type mice. Only after you breed these mice to another kind of mouse which express a pre-recombinase, either in neuron or in muscle. Most of these mice are commercially available from either Jackson lab or from some other labs. You breed this into a new mouse. This is the pre-log system. This is the pre-log site, which can be recognized by the pre-recombinase. This is the enzyme. Recognize these sites. Will cause recombination, loop out these two exons. So delete these two exons. This is the final product. We only have 3 exons left in the SOD1 gene. This will not be a functional SOD1. This will equal to knockout. So first, we knock out the SOD1 in the muscle. Use that strategy. This Western blot shows the expression of the SOD1 protein. In this MCAT is muscle-specific SOD1 knockout. You see the SOD1 is like more than 99% knocked down in this muscle. This is just a control gene. And in the brain, which is the most neurons, you see there's no decrease of the SOD1 protein. Indicates that SOD1 knockout is specific to the muscle. And this is the enzymatic activity of the SOD1. Shows the in the knockout mouse, the SOD1 activity is gone. This wild type still there, and in the brain, SOD1 activity still there, no change. So this tells us our model is good. We knock out the SOD1 specific in the muscle. We initially expect we knock out the SOD1 in the muscle only will lead to muscle atrophy. At least not comparable to the whole body knockout. At least you will see some decrease. However, to our surprise, we see that in this mouse, we see no change or even increase of the muscle mass. This is the muscle mass. This is the muscle specific knockout mouse. Increase the muscle mass compared to wild type. This is the absolute mass value in milligram. This is the percentage of the body weight. So either way, there's significant increase. However, there's only slight increase of the number of the fibers, and each individual fiber is the size of each individual fiber is similar, no significant difference between the knockout and the mouse. This shows a picture. There's some increase compared to the wild type. So this is completely against our initial hypothesis. What happened to this mouse? We look at why this mouse has larger muscle mass rather than smaller ones with hypothesis. Look at some of the growth signals. So initially we look at the IGF, AKT, mTOR signaling, which is the important growth signaling pathway in the muscle and in other tissues. So we look at the basically the activation of this pathway is the we look at the field the phosphorylation of the proteins involved in the signaling. In the knockout mouse, you'll see there's an increase of the AKT phosphorylation, also increase of the S6 phosphorylation. Both of them involved in the AKT mTOR signaling. Here shows the quantification of the Western blot. So indicates the loss of SOD1 in muscle leads to the increase of the growth signaling. So that's maybe part of the reason why this muscle, the muscle in SOD1, specific knockout the SOD1 mouse increased rather than decreased. And here, interestingly, we look further to the histology of the muscle fiber of the muscle of the knockout mice. To our surprise, we also find a very interesting phenomenon. This is the control, is the wild type muscle. This is the muscle specific SOD1 knockout muscle. There are dramatic increase of fibers with centrally localized nuclei. As you'll see the dramatic increase in two aging, for young and to middle ages. As you know, there are centrally localized nuclei in the fibers normally occurs only during injury, during differentiation, with immature fibers as centralized. They grow up from the satellite and then further mature, differentiate into mature. Once the fiber mature, all the nuclei will be in the squeeze to the membrane, under the membrane, to the side. So this is a very interesting phenomenon. We further look at the imaging of the the fiber and look at why there are so many central localized nuclei fibers. And we look at this wild type, it's typical. There's barely you can see. We sting the muscle fiber with 3 different antibodies. One is red, labeled is for the embryonic myosin heavy chain. These only appear in regenerative before immature myofibers. And green indicate the WG leptin, which is labeled the extracellular space. It's an extracellular matrix protein, which show the boundary of the fiber. And the blue color is the nuclei staining. In this wild type treated with the barium chloride, which damage the muscle, is a classic muscle regeneration, damage and regeneration agent. You'll see most of the regenerated fibers colocalize the EMHC and other centrally localized nuclei in this. However, in the mouse with knockout mouse, you only see very few of the central localized fibers express the EMHC. Majority of them do not express EMHC. Indicates they are not immature, undifferentiated fiber. So initially I thought this may be due to a dysfunctional differentiation. They stop because of the loss of the SOD1, some kind of damage of the signaling, stop the differentiation of the fiber, so the nuclei can stay in the central. So this type of fibers may have like abnormal function compared to the normal differentiated fibers. So that's next we'll look at the muscle function. So first we look at the individual fiber force. We just isolate the individual fiber and put on the machine which you attach to it. You test the maximum isometric force for each individual fiber. And here you see a dramatic decrease of the isometric max force in the compared to the wild type in the knockout mouse. That means the muscle, although the muscle mass does not loss, their force decreased. And we run these mice on the treadmill to see whether they have the overall physically fitness compared to the wild type. And this is the treadmill, and let the mouse run on the tread, and until exhaust, you see the muscle knockout mouse have a dramatic decrease of the time to stay on the treadmill. So they're another indicator for the decrease of the muscle function. We also put these mice on the root rod to test the motor neuron function, motor function. This test is more kind of to examine the motor neuron side of the function rather than the muscle side of the function. So in this test, we didn't see significant difference between the knockout and the wild type mice during the 3D test period. So that indicates the loss of the SO2 in the muscle, reduce the muscle function, but does not affect the motor neuron function. So after we look at all of this, we also look at why loss of SO2 didn't lead to muscle atrophy. Is there anything related to the oxidative stress, oxidative damage? So we look at the oxidative damage in this mouse model. We found there's no significant increase of oxidative stress in the MKO mice. So we look at the different genes expression in the muscle of MKO mice. These genes have been shown to be changed in whole body knockout mouse, but was not altered in the muscle specific knockout mouse. And we also look at the the hydrogen peroxide production in the muscle fiber. This is using isolated muscle fiber. Then you stain the muscle fiber, use DCF, which is the indicator of the hydrogen peroxide production. So in the whole body knockout mouse, DCF staining will show a dramatic increase in color changes. But in the MKL mice, we didn't show a significant increase of the DCF fluorescence. Although after an extended period of exposure, you will see there's a trend of increase, but it's not statistically significant. And we also look at the oxidation of the lipid, which is an indicator of oxygen damage. And we also didn't see any significant increase of the oxidation in the MKL mice. So this tells us there's no significant increase of oxygen damage in MKL mice. However, we cannot exclude the microenvironment changes of oxidative stress. And also we cannot exclude that the slight increase of ROS in MKL mice cannot be detected using this method. The slight increase of ROS may play an important role in cell signaling rather than cell damage. Most likely they can stimulate the cell signaling such as the AKT-mTOR signaling as I showed earlier, and some other pathways. So those the possibilities will be further explored in the future studies. In the first strategy, the second mouse model is to try to knock out SOD1 specifically in the neuron. So to see whether SOD1 knockout in neuron can change the muscle mass and function. So we cross the SOD1 flox mice, as talked about earlier, to the nesting pre-transgenic mice. These nesting pre-transgenic mice express the Cre-recombinase specifically in the neuron. After we cross these mice, we generate the neuron-specific SOD1 knockout mouse. This shows the Western, show that the we call it NKO. This is the whole body knockout. So you will see SOD1 is disappearing in every tissue, including muscle and brain. Whatever is there. In the neuron-specific knockout, you will see it's only knockout in the brain, which is neurons, mainly the neuronal tissue, but not in the muscle. And this is another model I won't talk about. It's neuron-specific transgenic, which all expressed in the neuron, in the SOD1 mouse. Our preliminary data indicate that neuronal specific knockout of SOD1 reduced the muscle mass at much late age compared to the whole body knockout. And there is a decline of the muscle function in the neural specific knockout mouse. And also there is a deterioration of neuromuscular junction in that mouse. This piece of evidence suggests that SOD1 loss, specifically in the neuron, plays a much more critical role in leading to the muscle loss in SOD1 mouse rather than the muscle fiber. To summarize, so the muscle-specific knockout SOD1 has no muscle atrophy, no significant increase of oxidative stress, and there are more fibers with centrally localized nuclei suggest either regeneration or abnormal nuclear fiber maturation. In the neuron-specific SOD1 knockout mouse, there is a muscle atrophy occurred much later, although much later, but it occurred compared to the muscle-specific knockout mouse. And there is a decrease of muscle function and also deterioration of neuromuscular junction. A mess. So,
You're okay. Got 10 minutes if you want to.
So the strategy number 2 is we try to restore the expression of SOD1 in SOD1 knockout mouse model. So try to see whether this can rescue the muscle function. So first we express the human SOD1 in the muscle, only in the neuron. with the neuron-specific promoter, synapsin promoter. After we generate the transgenic mice, we cross with the SOD1 whole body knockout mouse. After several generations of breeding, we get the mouse which has the SOD1, human SOD1 expressed only in the neuron, but not in the other part. The other tissues or cell types still lack of SOD1. So here shows the expression of the transgene and the internal endogenous gene. Here shows the SOD1 whole body knockout is disappear in every tissue, other tissues. This the gas muscle and nerve. Wild type endogenous expression in every tissue, of course. And the neuron-specific rescue SOD1 mouse, you see in the muscle, no expression because the human SOD1 specifically expressed in the neuron. So the muscle didn't see much. Even you can see a little bit that may be generated from the motor. And you will see there's a expression of the human SOD1 in the neuron, side nerve neurons.
They are molecularly different because
There-
of glycosylation or because of
The modification is similar. There's a little bit few nucleotide difference, and it's caused a dramatic shift of this. It's interesting. So we got the correct mouse model. So we'll test whether oxygen stress changed in the brain and also in muscle. And in the skeletal muscle, the gastrocs, you'll see this is the wild type, whole body knockout, neural rescue SOD1 mice. You see, even the muscle and in the brain, both tissues has dramatic decrease of the lipid oxidation compared to the whole body knockout. So this is very interesting because you rescue the SOD1 only in the neuron. You reduce the oxygen stress in the muscle. That's because of the talk between the motor neuron and the muscle. And the rescue of the SOD1 in the neuron also dramatically improve the neuromuscular junction in the rescue mice. Wild type is this is the neuron staining of the acetylcholine receptor. This is the muscle staining of the acetylcholine receptor. The wild type, you see the structure is intact. In the whole body knockout mice, disrupted, fragmented compared to the wild type. You see and this the overlay. You will see there some of them are not overlay and it's very fragmented compared to the wild type. However, in the neural rescue, the neural side, the plate, and the mouse side all improved compared to the whole body knockout. That indicates you rescue the SOD1 only in the neuron can improve the neuromuscular junction dramatically. Overexpression of the SOD1 in SOD1 knockout mouse in the neuron also improve the muscle function. Here shows the EPP is the end plate potential, and it's a miniature end plate potential. This isolated fiber, you have some electrode attached to that. We give some drug. You test the snaps, called end plate potential, which fire up the electric signals. You can use those electric to detect. So this is the whole body knockout. There's a very smaller reaction to the stimulation. The wild type and the neural rescue that improve dramatically. And also the miniature end plate potential also there's dramatic improvement compared to whole body knockout. And we also measure the function using the EMG method. This is the hind lamp. You stick to the motor neuron communication. You stick to the electrode to one end of the muscle, the other to the side nerve side to stimulate. You measure the propagation of the signal. So you measure like 5 times, and normally you have a precipitation if in normal mouse tissue. In the knockout you have a dramatic decline, like it stay there, it won't like decrease. And once you express the SO2, restore the SO2 in the neuron only in the SO mice, you back to very close to the wild type. Another indicator that neuron-specific restoration of SO2 improves the neuromuscular function. And furthermore, the expression of the SO2 in the neuron also improves the isometric force in the muscle fiber. You isolate the fiber, there is no motor neuron, and the fiber also has a much better function. So the force restored to the wild type. This is the hallmark of the muscle fiber.
Yiqiang, is the EPP connected to any specific ion channels or calcium or potassium?
That's basically the acetylcholine receptor.
So it's acetyl.
Yeah.
Okay.
Yeah, the calcium. This is more EPP is more calcium, and the action potential AP is more sodium.
Mm-hmm.
This is localized only in the synapses. For post-synapses, those signal is very short. So rather than the neuron, the muscle, after the EPP is with AP, action potential, change it to the sodium, to the muscle contraction.
Is this the bigger signal indicating there are more release of the acetylcholine from the neuron?
Yes, more healthy, more release. If you have a small peak, that means very weak, very small release. If you stimulate, very small. Normally, wild type like this peak, this is. Also, this miniature is like you consider the machine. You stick that, you use amplifier here. Those things. In the Hobart knockout, you rarely hear. That means very weak signal transduction in this Hobart knockout. To summarize on the part 2, that it's the reintroduction of the SO2 back into the neuron of the SO2 knockout mouse, reduces stress in muscle, also significantly improve the structure of neuromuscular junction and muscle function. To conclude, that oxidative stress and damage to both the muscle and neuron may be required to induce the muscle atrophy or sarcopenia in the SO2 knockout mouse model. The reason why, because if you knock out SO2 specifically in the neuron, you lead to muscle atrophy only in a very late stage, like after 20 months of age. Compared to the whole body knockout mouse, the muscle atrophy occur around 10 or 12 months of age. So although you didn't see any muscle loss in muscle specific knockout SO2. And however, the motor neuron seems to play a more important role compared to the muscle part. So there's multiple other important questions you need to address. And so the first one is why does the muscle specific loss SO2 leads to the muscle atrophy? So that's an important question need to be investigated. The second way is why the specific restoration of the SO2 in neuron leads to the restoration or the decrease of oxidative damage in the muscle part. So that means how the neuron communicate to the muscle in terms of oxidative stress or other things. And finally, what other cell types involved in the process? Because in each tissue, there, besides the neuron and muscle fiber, you have fibroblasts, adipocytes, inflammatory cells, and metabolic cells. So, and in the neuron part, you have astrocytes, microglia cells. All those may be involved in the process. So what we learn from this? So one of the part is exercise. The other part is proper nutrition or diet, as we show that color restriction reduce the damage and improve muscle function. And also, because we're interested in the oxidative stress of damage, so is there any possibility that antioxidants like vitamins can improve this? Some studies indicate that you take vitamins can improve muscle function. The last part is hormone supplementation. That has been widely, I think, used by some of the athletes, right? So one of them is the IGF, which is important in the muscle growth. So there is a mouse model called Schwarzenegger mouse. It's generated in Italy by a researcher, which expresses muscles with IGF-1, and the mouse has strong muscles. They call it Schwarzenegger mouse. Finally, I have to thank those people involved in these studies in the UTHSCSA Center, Holly, and Yunshi, and Yiqiang is here, and Aaron, and Dr. Richardson, and Yuji is involved in some pathology, and Carlos, I think is here, Carlos is in the geriatric. And also, some of the work shown here are from the former lab members, and they are both into postdocs in Harvard. And this is a big group project involving two other labs. One is in the University of Michigan, directed by Susan Booth. And the other lab is directed by Malcolm Jackson in University of Duke. It's funded by AIH, and we once. Yeah.
Thank you.
Thank you.
Thank you. Any
Nice.
final words? Any final questions? We were pretty short in time.
Yeah, just one. Are there a decrease of SOD1 in old human muscle, or
This is a disease.
is it?
Yeah, the only the ones the ALS, it doesn't increase rather than decrease.
Oh,
Yeah. Some of the mutations have increased, have decreased actually. Some of the mutations have maintained actually. But there is the increase of active stress. I think that would be normal.
Anybody measure the food consumption throughout the age with SOD about
Okay.
I just wonder whether the jaw muscle increases too.
Yeah.
They make a jaw effect.
Yeah, there are more questions than answers. Please.
Thank you.
Thank you.
thank you.
Thank you.
Good job.
That's our go all the way back there.
Thank you.